So, mobilization is the process by which these stored triglycerides within the adipose tissue and adipose cells are broken down into free fatty acids in the adipose cells. This process occurs primarily through lipolysis and catalyzes the hormone or enzyme HSL. Enzyme. Lipolysis is stimulated by hormones such as epinephrine, cortisol, and glucagon. These activate protein kinase A and other pathways that signal and phosphorylate HSL. Now, once activated, HSL will hydrolyze these triglycerides into free fatty acids, as well as glycerol. This will then be released into the bloodstream to transport into various tissues that need energy. Once it does that, after being released into the bloodstream, these free fatty acids will need to be transported to a targeted area or a targeted tissue like muscle or liver or something like that. And this will be catalyzed into energy production. But the FFAs, or the free fatty acids in the blood, are carried by albumins, a protein that binds 10 fatty acid molecules per albumin to itself. The albumin free fatty acid complex is very, very important for preventing these fatty acids from precipitating within the bloodstream, and it keeps them on track and going to the correct spot. Now, once they're delivered, cell uptake is to occur. Cellular uptake involves the transported FFAs to go from the bloodstream into the targeted cells, the muscle cells, or where they're going. This process is mediated by free fatty acids and free fatty acid proteins and CD36. This will facilitate the entry of FFAs across the plasma membrane into the cytoplasm, which is very, very important. Once it is inside the cell, the FFAs can either be oxidized or reduced in the mitochondria. This will generate ATP and esterify into triglycerides. So the difference between lipolysis in the adipose tissue and lipolysis in the muscle includes a couple of things. In the adipose tissue, lipolysis is regulated primarily by hormone signals and products like free fatty acids and glycerols that enter circulation between various tissues. Muscle lipolysis, on the other hand, tends to be more localized and relies on the breakdowns of IMTGs in order to provide energy for sustained physical activity or exercise. The muscle can store a small amount of triglycerides directly, which are broken down further in the body and within the muscle in order to catalyze and resynthesize ATP. Now, regulation of mobilization is a couple of things. Hormone signaling is a big part of regulation for mobilization. Hormones like epinephrine, norepinephrine, and glucagon activate hormone-sensitive lipases, or HSLs, through the CAMP, a lot of abbreviations, and PKA signaling pathways promoting lipolysis and promoting the release of free fatty acids. Insulin, on the other hand, is a key regulator of lipolysis, but this one inhibits lipolysis activity because it decreases CAMP levels by activating phosphodiesterase, and this will reduce PKA activity, ultimately decreasing HSL activity. For regulation of cell uptake, there are fatty acid proteins. Okay, fatty acid transport proteins. The fatty acid transport proteins and CD36s on the cell membrane will regulate the entry of free fatty acid cells going into the muscle cells, or into the cellular uptake part. There's also exercise and diet. Exercise increases the expression or the amount there is of CD36, as well as these fatty acid transport proteins. And this will enhance fat uptake for oxidation